So I'm going to speak about how a high-throughput screen led us to an unexpected discovery for the role of phase separation in the pathogenesis of Parkinson's disease. So Parkinson's disease is a common movement disorder, which is characterized by the accumulation of a protein called α-synuclein within neurons in the brains of the patients. And this protein α-synuclein forms round inclusions that are called Lewy bodies. So in the early stages of the disease, the Lewy bodies first appear in the bottom part of the brain, which is called the brainstem. However, as the disease progresses, the Lewy bodies also appear higher up in the brain, and eventually, in the end stages of the disease, involve the cortex. So it has been hypothesized that this apparent spread in pathology is underlined by the pathological cell-to-cell propagation or transfer of α-synuclein. Therefore, it is important to understand the mechanism with which cell-to-cell propagation of synuclein is happening. To achieve this, during my postdoc that was funded by an HFSP fellowship, we undertook a high-throughput screen for α-synuclein propagation. And the first step to a successful high-throughput screen is to have a robust model system in tissue culture. So to achieve this, we cloned a construct encoding GFP to a α-synuclein RFP fusion protein. So when we transfected cells with this construct and it's then transcribed and translated into a protein, the protein is immediately cleaved at the 2A position, leading to the production of two independent proteins, GFP and α-synuclein RFP fusion protein. The latter is then able to transfer to neighboring cells to propagate. And we can tell apart the two cell populations based on their color. The cells that are positive for green and red express synuclein through transfection, whereas the cells that are positive only for red but not green have taken up synuclein through propagation. And then we did a high-throughput screen with this assay, imaging-based, in 384 well plates. And to cut the long story short, after a long iteration, we ended up with a final list of 38 genes, whose knockdown either increases synuclein propagation or decreases synuclein propagation. Two of those genes were of particular interest: TAX1BP1, which is an autophagy receptor, which has also been found to be involved in the mitophagy in Parkinson's disease. And the other gene is ADAMTS19, which has been found to be involved in the proteolytic cleavage of α-synuclein. To start studying the mechanism of action of those genes, we hypothesized that they are involved also in the formation of α-synuclein inclusions, as I previously mentioned, the Lewy bodies. So the next challenge was to model α-synuclein inclusions in tissue culture. And we did this based on a recently proposed model for how synuclein dysfunctions leading to synuclein inclusions. And this is a tetramer disruption model. According to this model that was developed in Dennis Selko's lab, α-synuclein physiologically exists in tetramers within the cells. And it being an intrinsically disordered protein, the tetramers are essential to keep it sequestered and under control. However, in Parkinson's disease, these tetramers fall apart, which leads to an excess of synuclein monomers. And then the synuclein monomers being disordered, they can propagate, aggregate, dysfunction, and this is a toxic feature for the cells. This model fits in beautifully with the genetics of Parkinson's disease, as Mendelian mutations in synuclein cluster around the protein regions that are thought to be important for the formation of tetramers. And Dennis Selko's group developed a cell line to model this process in tissue culture. So α-synuclein has 5 repeat domains in its sequence, each comprising of 6 amino acids. So they mutated strategically 3 amino acids, glutamic acids, into lysines. And they found that this mutated synuclein, the triple mutant, when they transfect cells with a plasmid encoding it, it spontaneously forms synuclein inclusions that look a lot like Lewy bodies. So we got this cell line from them, and we knocked down TAX1BP1 and ADAMTS19. And we found that this knockdown results in a statistically significant increase in the number and decrease in the size of the synuclein inclusions. We also found that this knockdown does not modify the total amount of synuclein per cell, as quantified through flow cytometry. So this initial finding served as an initiating point to dissect the mechanism with which we go from tetrameric synuclein to inclusions and finally to cell death. So here's a close-up of the synuclein inclusions, and to me those looked a bit like droplets. And since phase separation is quite fashionable at the moment, we decided to study if these form through phase separation. And to do this, we undertook fluorescence recovery after photobleaching experiments, or FRAP. Indeed, we found that the synuclein inclusions have a large spread in their immobile fraction, which ranges from between 20% to 80%, with the average being around 36%, which means that largely synuclein inclusions are liquid. And this supports that they are liquid condensates that form through phase separation. We also did a time course experiment in which we did FRAP on the synuclein inclusions right at the point where they were formed, 16 hours after doxycycline induction of the cell line. And we found that these baby inclusions had a wide spread in terms of their liquid versus solid status. Some of them were 100% liquid, some of them were 100% solid. And this made us conclude that synuclein inclusions initially form either through phase separation or through aggregation or through a mix. However, as time progressed, the liquid fraction of the inclusions dramatically increased. And this suggests that while synuclein inclusions form through a combination of methods, they do expand in size through phase separation. Supporting the role of phase separation in the formation of synuclein inclusions was their shape. So synuclein inclusions as quantified by the circularity index were quite round, and this supports them being droplets. Finally, we knocked down our two genes of interest and found that when we knock down ADAMTS19, it increases the liquid fraction of synuclein inclusions. And this supports that this gene, when we knock it down, it makes the inclusions more dynamic, and therefore they can interact with each other with fusion or fission, which probably makes them be more small, as we saw in the previous slide. So now the next question is, what comes first? Is it tetramer disruption or phase separation? So it has been found that a protein that has a higher quantity of lysine has a higher propensity to phase separate. To check whether the 3K mutant first induces phase separation or tetramer disruption, we used the intact wild-type sequence of the protein, and we added 3 lysines right before the repeat domains, but we kept the repeat domains intact. So we hypothesized that if phase separation comes first due to the higher quantity of lysines, then this mutant synuclein would again form synuclein inclusion spontaneously. However, this turned out to not be the case. This protein behaved as the wild-type protein as seen through imaging, which means that what comes first in the 3K synuclein is a disruption of the synuclein tetramer, followed by phase separation. We then plotted the disorder of the 3K synuclein and the wild-type synuclein. Surprisingly, we found that the 3K synuclein is less disordered than wild-type. And this appears to contradict our experimental results, because we know that disordered proteins have a higher propensity to phase separate. So we thought that there might be an additional step going on. So now what is this additional step? We looked at the electrostatic charges across wild-type and 3K synuclein. And we found that in the region of the hairpin that is supposed to form the binding pocket in the tetramer and to interact with each other in the monomer, we found that this is positively charged in the wild-type protein. However, it's more positively charged in the 3K mutant. And we hypothesized that lipid binding might have a role in modulating the phase separation behavior of alpha-synuclein. And we are currently studying this through molecular dynamic simulations for the association between synuclein and specifically phospholipid bilayer membranes and phospholipids. This analysis is currently in progress. So since we got interested in lipids, and since we know that the brain is the second most lipid-rich organ after the adipose tissue, we wanted to simulate a lipid-rich environment in tissue culture. To achieve this, we treated the cells with a compound called oleic acid, which increases the contents of neutral lipids in the cells that form a higher number of lipid droplets. Surprisingly, we found that these lipid droplets embed themselves within synuclein inclusions, and they make them have a Swiss cheese appearance. When we knock down TAX1BP1, this relationship between lipid droplets and synuclein changes. So we have a big lipid droplet, a single big lipid droplet that is surrounded by a thin rim of alpha-synuclein. We also wanted again to study the molecular grammar of synuclein. So we took the 3K sequence, and in the repeat domains, there is a valine. And the valine is a hydrophobic amino acid. So we mutated it to a completely opposite, glutamic acid. We hypothesized that if the association with lipids is mediated by valine, and it's integral in the entire process between tetramer and inclusions, we thought that this mutation would completely affect this entire process. And indeed, we found that this valine to glutamic acid mutant is unable to form tetramer, to form inclusions, and it's also unable to associate with lipid droplets. We also have some preliminary data showing that knockdown of TAX1BP1 results in increased reactive oxygen species in the cytoplasm. And this could be the mechanism resulting to cell death in this system. I'm not showing this data yet because it's not finalized quite yet. So now, how does all this fit into the genetic basis of Parkinson's disease? So going back to our 38 genes we identified through the propagation screen, we used a gene expression analysis technique called weighted gene co-expression network analysis. So this analysis technique groups genes, their expression levels, based on pairwise correlation coefficients into modules, gene expression modules. And it has been found that the genes that end up in the same modules are functionally related. And this analysis has been done, for example, for Alzheimer's disease and other neurological diseases. And we found that indeed our 38 genes cluster within gene expression modules in several regions of the brain that are involved in Parkinson's disease. And also, these gene expression modules are enriched in expression signatures shown in neurons. And as we know, neurons are the cell type that is most severely affected in Parkinson's disease. Then looking at the disorder of those 38 genes, we looked separately at the upregulators and the downregulators of synuclein propagation. And we found that while the upregulators are similarly disordered as the entire proteome, the downregulators are different. So they have a reduced amount of disorder as seen in the probability plot. So this observation made us expand this analysis on a wider scale. So we looked at the disorder of all the proteins that are expressed in each and every one of the 13 regions of the brain that we studied. And we plotted the disorder using a probability plot. When looking at all those genes, we didn't see a difference in the probability plot compared to the proteome. However, when we focused on just the neighboring genes of TAX1BP1 and ADAMTS19, the picture was completely different. We observed a beautiful gradient for the various brain regions, which was particularly pronounced for TAX1BP1. So the neighboring genes of TAX1BP1 were more disordered within two regions that are critical for the pathogenesis of Parkinson's disease, the putamen and the caudate nucleus, which are the basal ganglia. And so this finding indicates that a common mechanism underlying the pathogenesis in Parkinson's disease and the dysregulation of α-synuclein could be the dysregulation of phase separation on a larger scale. So in conclusion, we have shown that the combination of a high-throughput screen for a biological mechanism, together with orthogonal approaches, including gene expression analysis in human brains and follow-up experiments with cell biology and structural analysis and genetics, is a powerful approach to understand the molecular basis of complex diseases. We also have indications that α-synuclein follows a strict molecular grammar, and small changes in the sequence can completely affect its biophysical behavior. We also have indications that dysregulation of disordered proteins on a wider level could be contributing to the pathogenesis of Parkinson's disease. And finally, we have outlined a skeleton pathway suggesting how we get from the tetramers to inclusions and to final cell death, and how propagation of synuclein integrates within this pathway, and how genetic networks in Parkinson's disease are involved in this process. And I would like to thank my lab at Rutgers, which opened in September, and everybody has worked very hard and has done a great job in moving this project forward. I would also like to thank HFSP that funded my long-term fellowship, during which I completed the high-throughput screen that formed the basis of this study. And finally, I would like to thank our collaborators at Rutgers, UCL, and Harvard, and my previous mentors with whom I completed this high-throughput screen, and they were kind enough to allow me to take this work with me to continue it in my lab at Rutgers. Thank you for your attention, and I would be happy to take any questions.
Thank you, Eleanna.
You're welcome.
Questions, please.
Oh, thank you for a really cool talk. My question is that to track your protein, you fuse the GFP tag, and I'm just wondering that this fusion of GFP to your α-synuclein has any effect on the biophysical property of liquid condensates like surface tension, partition coefficient, or like diffusion, or other things?
Yeah, thank you for this question. Indeed, it's a common concern that when you add a tag to a protein, especially a large tag to an amyloidogenic protein such as synuclein, it can affect its biophysical behavior. So we have not looked specifically into the condensates that form with tagged synuclein versus untagged synuclein. We are in the process of doing this using recombinant synuclein in vitro. However, I would say that the biggest finding that is reassuring in our case, that the behavior is not significantly altered, is that our findings fit in with the genetic basis of Parkinson's disease. And as we know, the genetics is the ground truth. So the fact that this mechanism fits in with the genetics and the genetic networks underlying the disease is reassuring that what we are seeing in cells recapitulates with quite some accuracy what's happening in reality in the brain.
Very interesting talk. So you said that there is the intermediate step where there is like interaction with the phospholipid bilayer for the phase separation, right? Mm-hmm. Have you tested experimentally or with the simulations interaction with synaptic vesicles?
No, we have not done this yet. It would be interesting to do because, vesicles are also lipid-like, yes, surrounded. Actually, what we have done is we have done electron microscopy, CLEM analysis on the synuclein inclusion. So we lysated the cells with hypotonic lysis buffer, and then we did electron microscopy in the inclusions, and we found that they're actually rich in vesicles, and that synuclein directly binds upon those vesicles. So I think that what you propose is a promising experiment, and our findings could also have a role, like if we expand them, we could get more insight into the synaptic role of synuclein and synaptic vesicles.
Yes.
Thank you.
Hi. Very nice work.
Thank you.
You probably replied already partially to my question, but I would like to ask it anyway. Do you have any evidence that this is happening in vivo? And do you know whether different kind of neurons are differentially susceptible to this phenomenon? Because of course, like dopaminergic neuron in the substantia nigra, they are in the basal ganglia are particularly important in Parkinson.
Yeah, so this is a good question. So for the in vivo, so the thing with synuclein inclusions is that we don't have any really good model systems for them. So in my opinion, the tetramers has been a breakthrough because what we get, the inclusions that we get, recapitulate what is seen actually in the Lewy bodies. So they have done a mouse model for this tetramer disruption system. We have not studied our genes of interest in that model system. It's in the plan, like medium-term plan, but we have not done it yet. In terms of the specific types of neurons that are affected, we don't have any data yet. We do have dopaminergic neurons in the lab derived from stem cells from patients with triplications of synuclein versus wild type, like non-mutated healthy people. And we are going to look at those pathways in those stem cell-derived neurons, dopaminergic neurons in the lab, to see if it's more specific. We'll also look at glutamatergic neurons, the eye neuron system, to see if we see a difference between the dopaminergic neurons and the glutamatergic neurons, if we see a difference depending on the cell type.
Thanks.
You're welcome.
Okay. Thank you. I don't see any more questions, so thank you very much, Eleanna.
Thank you.